Imagine reading a headline that says, "Pancreatic cancer eliminated." If you or someone you love has ever faced pancreatic cancer, your heart probably jumps, because pancreatic ductal adenocarcinoma, or PDAC, is one of the toughest cancers we know. Now, in early 2026, news spread about a research team led by Mariano Barbacid at Spain's National Cancer Research Center, CNIO, reporting something that sounds almost unreal. They eliminated pancreatic tumors in mice in a way that didn't allow the cancer to come back through drug resistance, at least within the time they observed. So, is this a cure? Is the end of pancreatic cancer finally here? Today, we'll break down clearly and honestly what Barbacid's group discovered, why it's scientifically exciting, and why it does not mean we have a cure for human PDAC, at least not yet. PDAC is the most common form of pancreatic cancer. It's notorious for three big reasons. That combination, deep responses plus no obvious resistance plus tolerability in mice, is why people are calling it a breakthrough. Let's give credit where it's due. This work matters for science in several ways. A. It strengthens a core cancer lesson. Combination therapy beats single hits. In infectious diseases like HIV or tuberculosis, doctors learned long ago that single drug therapy often fails because resistance emerges. Combination therapy became the model. Cancer is similar. When you hit one target, evolution and selection can favor resistant cells. A rational combination strategy can make resistance much harder. This study is an example of that logic executed in a very targeted way. Block multiple critical nodes in the KRAS-driven machinery and the pathways KRAS controls. For a long time, KRAS was considered undruggable, not because scientists didn't want to drug it, but because it was extremely difficult to hit safely and effectively. In recent years, KRAS-targeting drugs and related pathway inhibitors have finally begun to appear, and that's a genuine turning point. But there's a huge catch: resistance. Here's the key idea. Cancer is not a single enemy soldier. It's more like a shifting army. If you block one route, it may open another route. If you cut one wire, it may reroute the circuit. This is exactly what happens in many PDAC treatments. A drug blocks one signaling pathway, tumors shrink or slow down, and then cancer cells adapt and become resistant, so the tumor grows again. Barbacid's team focused on that specific pain point. How do we stop resistance from appearing? The CNIO team's strategy can be summarized like this. Don't block KRAS signaling at just one point. Block it at 3 points, so the tumor has far fewer escape routes, skin issues, gastrointestinal effects, and more. So well tolerated in mice is encouraging, but it's not proof of safety in humans. Reason 2. Human PDAC is intensely heterogeneous, yet their tumors can be biologically very different. Different KRAS variants, different co-mutations, different microenvironments, different immune interactions. In the life science reporting, the researchers explicitly point out that patients can have many alterations, and they plan to test additional models to reflect this diversity. A combination that works across several models is promising, but it still doesn't guarantee it works across the true messy variety seen in clinics. Reason 3. Many therapies work on primary tumors but fail against metastases. In real life, PDAC often becomes deadly because it spreads. Metastatic disease brings extra barriers, different organ environments, different drug penetration issues, and often different biology. A mouse model can simulate some of this, but not perfectly. Reason 4. Drug delivery and dosing are hard in real patients. In the lab, you can optimize timing, dosing, and control variables tightly. In patients, you must deal with age and frailty, liver and kidney function, other medications, and the practical reality that PDAC patients can become weak quickly. A triple therapy may be powerful, but can patients tolerate it? Can it be given safely in the real world? Those answers require careful trials. Reason 5. No resistance observed is not the same as resistance is impossible. In studies, no resistance developed means, within the time frame, with those conditions, the tumors did not regrow in the ways researchers looked for. In humans, resistance can take longer, appear through different mechanisms, or emerge in tiny subclones that weren't captured. So the correct translation is, this strategy may suppress common resistance routes in these models. Not resistance is solved forever. Reason 6. The researchers themselves say clinical trials are not ready yet. This point is easy to miss when headlines get emotional. Barbacid is quoted as emphasizing they are not yet in a position to carry out clinical trials with this triple therapy, and that optimizing it for clinical use will not be easy. That's not pessimism, that's scientific responsibility. If you want to understand why cures take time, here's the typical pipeline. 1. Reproducibility. Other labs try similar approaches and confirm results. 2. Optimization. Find the best drug versions, doses, schedules, and biomarkers. 3. Preclinical safety studies, toxicology, interactions, long-term effects. 4. Phase 1 trials. Is it safe in humans? What dose can we use? 5. Phase 2 trials. Does it show activity in a specific patient group? 6. Phase 3 trials. Compare against standard treatments to prove benefit. At any step, the approach can fail, not because scientists are incompetent, but because biology is difficult. Still, when a study offers a clear mechanism-based strategy to prevent resistance, that can accelerate the process because it tells the next generation of trials where to focus. Let's land this plane with a balanced message. This work is exciting because it suggests a principled way to overcome one of PDAC's biggest problems, resistance. It achieved unusually deep responses in multiple models, and it offers a clear biological story to test in humans. But it is not a cure because it's not proven in humans. Human PDAC is more diverse and clinically complicated, and we don't yet know whether the approach can be delivered safely and effectively to real patients. So if you see headlines saying pancreatic cancer cured, the more accurate translation is, a promising combination strategy eliminated tumors in mice and may guide future therapies, but human trials are needed. That is still big, that is still progress, just not the final chapter.